Now, next we have the flash talks for the poster presentations. So before we start those, if all of the presenters can come and stand or sit somewhere near the front, then we can quickly go through. Okay, well, the first one is mine, so I'm in the right place. Yeah, is everyone else who's presenting ready to come up afterwards? Okay. So, yeah, I'm a postdoc at ANU. I work with Justin Borovitz, and I wanted to quickly tell you about a project that I started this year. The aim of the project, well, it's really motivated by global forest dieback. So across the world, as in Southeastern Australia, many forest ecosystems are experiencing demographic decline from combinations of abiotic and biotic stress. Often it's complicated and poorly understood. One of the hopes is that if we can phenotype individuals across forests, looking at canopy traits and canopy conditions, to get a look at the variation in stress responses, we could then go down onto the ground, sequence the genomes of selected individuals, and try to tease apart the environmental and genetic determinants of stress response. So we haven't got there yet with this project, but we're working towards that. And we're at a stage where we can start looking at individual tree time series. And I'll show you a pipeline that we've developed towards doing that. So here we've got LiDAR flight data from across the entire ACT, which is a few thousand kilometers squared. So this is 18 points per meter. And with that, we've been able to segment the tree crowns of 5 million large trees. An example of that is shown in that top middle plot. So for each of those, well, we're fortunate that 100,000 of those tree crowns we can assign species labels to through some field surveys that were conducted. So we have this. So next, we actually extract PlanetScope CubeSat data, which is high-resolution imagery almost every day with 8 bands. So we've got this time series for every single tree, multi-band. And that's really nice to start exploring species classification. For an example, I've shown some summaries of time series for oak species that are planted around Canberra and 3 eucalyptus species. So you can see the variation between these groups. Obviously, oaks lose their leaves, but we're really more interested in the variation within species. So to what extent is this caused by genetic variation, environmental variation, and just noise in the data? And so that's kind of what I'm interested in talking about today. If anyone is interested in phenotyping trees using space, come and find me. My poster was actually yesterday, but I'd be really happy to talk to you all about it. Thanks.
Good morning. I'm Etsushi KUMAGAI from Nara, Japan. It's an honor to be present our work today. Biomass and leaf area index are key factor for crop yield. Typically measured by destructive sampling method, like this. Like this picture. So which are time consuming and labor intensive. To address this, we use hyperspectral reflectance as a non-destructive and faster alternative. Widely used full spectral band model like partial least square regression as a gold standard, but can lead data redundancy. So we try to apply the band selection technique to streamline process and improve the prediction accuracy. We collected the time series data of canopy scale, reflectance, biomass, and leaf area from many soybean genotypes across many years. Addition to PLS, and we also evaluate two band selection technique, bootstrap lasso and ensemble running. These methods allowed us to select the most important one for accuracy. This figure shows the PLS using all spectral band shows the highest accuracy for area predicting. However, some running and bootstrap lasso using a fear band performed well. We conclude that both bootstrap lasso and answer running present efficient alternative of PLS by simplifying data complexity and maintain prediction accuracy for LAI. This selected band for prediction LAI could contribute to advances in the multispectral sensor for agriculture monitoring, I believe. Thank you for your attention.
Hello, everyone. So microRNA seem to be a very new term and important term right now because of the news. The Nobel Prize for 2024 is given for the discovery of microRNAs, and microRNA play a very crucial role in my analysis. And so here, basically what we did was, so the questions that come forward in my analysis is, what are those genes that regulate the flowering time in sorghum? And flowering time being a very important trait, because it affects the yield and a lot of other things. So we are here trying to understand what are those genes that are affecting flowering time, what is the pathway that regulates flowering time, and how different is sorghum when compared to other grasses like maize. So here we are taking a very big population of 900 genotypes, and this population we collected the phenotypic data and scored flowering time for it. And we collected tissue data that was collected only in like 2 hours for this complete field by my lab mates. So first picture shows the diversity of sorghum that we had, and second picture shows that I was collecting the tissue. And yeah, so 2 hours was a very small time because RNA seems to change with time. And next is these tissues that were collected were then, we created a gene expression for it. I done TWAS analysis, and a lot of genes that we can see here, the FT, the MADBOX genes, and these genes are actually regulating the flowering time in sorghum. So the next thing is, what is that pathway that regulates flowering time? So here it is the microRNA that is actually playing a very crucial role in the aging pathway of sorghum. And if you want to know more about this, please come and visit my poster, which is 52. I would love to talk about it. Thank you so much.
Hello, everybody. My name is Mildred Mayorga. I am a researcher at the Alliance Biodiversity and International Center for Tropical Agriculture. Okay. Pasture holds significant potential for sequestering carbon in soil due to their extensive coverage and their ability to allocate a substantial part of photoassimilate below ground through their root systems. Deep roots are an interest and particular interest because they can transport organic carbon to soil layers where it remains more stable. So, with the objective to identify tropical forest grass genotypes with root traits that promote soil organic carbon accumulation, an experimental trial was conducted involving root phenotyping of 180 genotypes grown in cylinders filled with soil. The evaluated genotypes included accession and hybrids of 5 different species of tropical forest grasses. Root phenotyping was performed using root images that were segmented and processed using a root image analysis software to quantify morphological traits. Additionally, we evaluated biomass distribution and an indicator of root depth. We identify 22 top-performing genotypes clustered by root traits, demonstrating increased root length, area, volume, and high root-to-shoot ratio, and deeper root systems. Thank you.
Greetings, everyone. I'm Sai Subhash, currently doing complex biosystems, PhD in complex biosystems from University of Nebraska-Lincoln. I'm currently working with Dr. Jinliang Yang. Our lab focuses mainly on quantitative genetics and population genomics. Today, I'm going to talk about nitrogen use efficiency in sorghum. Nitrogen is the most essential macronutrient often supplied for enhancing the grain yields. It is the most expensive and energy-intensive input for global agriculture production. However, cereals are inherently inefficient in uptaking this nitrogen, so excess amount of nitrogen causes severe environmental issues and also economic concerns. So I'm working with sorghum. Sorghum is the fifth most cultivated cereal and also a multi-purpose commodity crop. Despite being the C4 nature, it requires high amount of inorganic nitrogen fertilizers for higher yields. So for our study, we have grown 346 accessions from Sorghum Association panel, which were phenotyped under high nitrogen and low nitrogen conditions. The first picture is the data that we collected from the greenhouse and also as well as from the field. We have much more data within more years and all. I have analyzed greenhouse traits here. I'm showing the 4 different traits, which is plant height, leaf count, seedling fresh weight, and seedling dry weight. X-axis is the unit of measurement here, and y-axis is the density. So we can clearly see that there is a variation for all these traits between the treatment conditions, between high nitrogen and low nitrogen conditions. So subsequently, we have used high-density genomic marker data set obtained from whole genome sequencing and conducted genome-wide association studies. We have obtained pretty much previously well-defined genes and also the novel genes by using this updated marker data set. We have updated with reference genome of version 5 in sorghum, and which is highly accurate, and we are happy to share our data with anyone who is working with sorghum, especially with the Sorghum Association panel. And my main objective of this study is to identify the genetic components underlying for different nitrogen responses. And if you want to talk more about my research or maybe about my data regarding the Sorghum Association panel, I'll be around. Actually, my poster was until yesterday or until this noon. My poster number is 48 if you want to discuss and see my poster as well. Thank you.
Hi, my name is Liz Popowski. I'm with the Kiwifruit Breeding Center based in New Zealand. Now, we are not implementing high-throughput phenotyping systems in the orchard yet, but that's why I'm here, because I'd love to get some really good ideas from you. So what I do want to share, though, is before we undertake this process, we wanted to understand what are the traits that we need to measure for yield components. So first, a little bit about how kiwifruit grow and produce flowers. What are the methods that we are currently using to assess yield, and how do they compare? And finally, future directions. So kiwifruit produce fruit on 1-year-old canes. We typically grow them on a pergola structure. So that photo is us looking up into the canopy after winter pruning. And what I want to point out is that there's variation in the size and length of the canes for each of the vines. The vines are planted about 750 millimeters apart, so they're fairly close. And also that canopy structure changes every year as you remove the cane and tie down the next one. So the 4 different methods we're going to talk about is the total number per vine. So that is just the fruit count per vine irrespective of its size. Then we'll go along with the second one and calculate fruit per linear meter by measuring the canes that only have fruit on them. The third method that we talk about is a system we call cane mapper, which gives us an allocation of where the shoots are located, whether they're floral or vegetative, and how many inflorescent groups per shoot. And the last one is the components of yield, which gives us everything that cane mapper does, but also helps us understand where we're missing yield potential with winter buds that have not produced shoots. So when we start comparing them, looking at the raw data, there's a fairly decent correlation between the total fruit number and fruit per linear meter. However, what we're interested in is a particular yield band for the growers, and that's highlighted in the red box. When we take each of the methods and go, well, how do the rankings compare between the total fruit number and fruit per linear meter? Across the whole spread, there's a really good correlation. But when we look at just what's in that red box, the correlation falls apart. Thus, total fruit number per vine is not going to be the ideal solution, and we're going to need to collect more information. This is where the next two systems come in, looking at cane mapper and components of yield. All three of these genotypes that we're looking at have the same number of fruit per linear meter, but a very different architecture type. So in the first one, even distribution of shoots and fruit number per shoot. However, as you go through, they end up with just at the apical end, which is a challenge we have in kiwi fruit. And looking at a more detailed method, this is a photo of a winter bun. You can see one of them on the left-hand side has started to initiate a shoot. Well, the one on the right-hand side is not going to initiate a shoot, and that's an area where we're losing potential. So what method you want to do is going to depend on the question and also the amount of resources that you have available. We would like to go forward and start to do some kind of high throughput system in the orchard, knowing that our challenge is that we are looking up into the canopy. So if you'd like to know more, please see me afterwards, and thank you for your attention.